Welcome back. I'm Ed Highland, and this is DDW TV. We are here in San Diego for Digestive Disease Week, and we're glad you're able to join us. We're also very glad to hear from our friends at Ferring Pharmaceuticals. Ken Blount is here. Some very exciting news coming out, and we were just chatting before we started this actual interview, and this is incredibly exciting. We're talking about RBX 2660. Tell us about it, how it came about, and what you're doing with it.
Sure, so RBX 2660, it's what the FDA calls a live biotherapeutic product. And what that means is it's an investigational drug product that's comprised of live bacteria. And the reason it is because the bacteria in our gut, what we call the microbiome, can get disrupted by a lot of things like antibiotics. And they need correction, they need restoration, if you will. And that's why we've developed these investigational live biotherapeutics. The current one is RBX 2660 that we're developing.
Now, for a layperson like myself, tell us how this perhaps differs from, we talked about probiotics before, but more recently, fecal transplant has been what has been generating a great deal of news.
Sure, so a fecal transplant is what it sounds like. It's where you take a stool sample, you process it into a suspension, and you administer it to a patient. The goal is to correct or restore the microbiome. However, it's not an approved treatment. It's not FDA approved at all. It's also not standardized. So what you get as an FMT or a fecal transplant in San Diego may be very different than what you could get in Boston or New York. There is no standardization. There is no requirement to do it a specific way. The amount of bacteria that you're administering could be different. The pathogens that you screen to ensure are not there could be different. It's not standardized. So when we make a live biotherapeutic product, that means we've worked directly with the FDA and their guidelines for how to standardize. And that means also that whenever a patient gets a dose of this investigational treatment, they have an assurance that the FDA has looked at it and that there are certain guarantees of standardization in every single dose. And that's really the key difference between them.
And again, we need to emphasize this is investigational live biotherapeutic. Give us some thoughts, if you would, on sort of where you go from here. I believe you mentioned earlier that you were expanding a little bit as part of the fill me in on the blank.
Sure, so we've run now 5 large clinical trials. The most recent of these was a large phase 3 trial that was randomized, double-blinded, placebo-controlled trial, and that simply is the gold standard for generating data that says this could be safe and it could be efficacious for patients that are being treated. That's the sort of end, if you will, of our large pivotal data demonstration. Now we do still have an ongoing phase 3 clinical trial, and that's really aimed at continuing to see how could this work in a real world patient population, not just a very controlled clinical trial population, but a real world practice population. That is still ongoing, but the bulk of our data has been completed, and we are interacting with the FDA as we have for a while, really aimed at the first approval in this space.
Can you get real detailed for us and talk a bit more about the microbiome and the bile acid replacement?
Sure, so the poster that we really presented here, that was a poster of distinction, was really aimed at looking at 3 clinical trials that we've run. We already know the clinical outcome, we've presented that at DDW, but this was focused on what happens to the patient's microbiome and their bile acids in their gut from before to after the investigational treatment. And what we found was a clear restoration from a very unhealthy state before investigational treatment to a healthier state after. And what I mean by that is there are certain classes of bacteria that we know are associated with healthy individuals. Those were depleted in our patients before the investigational treatment. They were increased after treatment. Similar with the bile acids. So the indication we're treating, in particular, or the indication that we're investigating, is called Clostridioides difficile infection. And it's an infection that happens in the gut. When you have an antibiotic, it can disrupt your gut bacteria and allow this pathogen to grow, cause severe diarrhea. It's a debilitating, potentially deadly disease. The real challenge is that it's treated by antibiotics. So the standard of care is the same thing that can cause more problem to the microbiome. So what we're aiming to do is restore that microbiome after the antibiotic treatment. That's been the essence of our clinical trial program. Our clinical trials are aimed at reducing the recurrence of C. difficile after a standard antibiotic treatment. Now the bile acids are important Our gut and our liver produce what are called primary bile acids. Those are converted only by a healthy microbiome into secondary bile acids. Primary bile acids help C. diff grow. You don't want those to be the biggest part of your gut. If you have a healthy microbiota, it can convert those to secondary, which actually repress C. difficile and can actually repress the likelihood that you will recur. So what we saw in these trials that we've shown today is that when a patient comes into our clinical trial, they actually have mostly those less healthy primary bile acids. And then after treatment and continuing on to the end of our observation period, they shift to more of the secondary that repress C. diff. So it's very consistent with the microbiome data that we saw, in which you see a less healthy going to a more healthy state from before to after the investigational treatment.
I know you can't talk about time frames, but this is a very exciting time, and I know we're in the very near future going to have some wonderful options to present for clinicians out there to be able to use in their practices. Thank you so much for coming by, and I assume folks can stop by the Ferring booth as well on the show floor.
Absolutely, and we'll be around the entire conference.
All right. Ken, thank you so much for coming by and joining us. And this is DDW TV. Thank you for joining us.